Stéphane, thank you very much, and also Lionel, to invite me to this meeting. The purpose of my talk today, it in fact a use case about Bayesian generative artificial intelligence framework, and the idea was to apply it in order to facilitate modeling genetic susceptibility in prostate cancer. The idea was how to perform, integrate a new tool with generative AI in the Bayesian network to study this problem. First of all, I would like just to introduce the context, to give you the context of this use case. The context is prostate cancer susceptibility. Why? Because prostate cancer is one of the most common cancer diagnosed in the world, and also obviously in France, as you can see on this slide. But also because mortality from prostate cancer is also a problem. The main problem usually is lung cancer, but prostate cancer is the second cause of death in high-level communities or countries. The diagnosis of prostate cancer currently is based on a blood marker, prostate-specific antigen. This blood marker, if it was elevated, we perform imaging using a magnetic resonance imaging and in biopsy to confirm the diagnosis. So the risk of prostate cancer is multifactorial. A large part of this risk is attributable to age, but also with genetic susceptibility. Genetic susceptibility is present with familial history of cancer, obviously prostate cancer, but also other cancers, such as breast cancer, and also ancestry, because patients with African or Caribbean ancestry have an increase of risk of prostate cancer related to genetic factors. Environmental factors such as obesity or other nutritional factor could be accounting about 30%. But at this time, these environmental or ecological factors are relatively not very well understood. The problem of prostate cancer screening was debated during the last 20 years, because prostate cancer screening of prostate cancer based on the prostatic specific antigen tool, blood marker, is not very useful, in fact, or efficient, because you can detect a small tumor, which will be not dangerous for the life of the patient, and create over treatment. And on the other hand, this marker seems not to be reduce significantly the deaths and the population. So recently, it has been shown that the use of targeted screening based on genetic factor could increase the efficiency of prostate cancer screening and reduce the deaths in different countries. And in Europe, a new program based on targeted prostate cancer screening have been launched. So today I would like to just show you how it is possible to estimate the importance of a genetic factor and a polygenetic factor in the prostate cancer risk. During the last years, since 10 years ago, susceptibility of prostate cancer have been explored with specific markers. These markers are the single nucleotide polymorphism, and we have this small change in your DNA sequence between different population and patient, and some of these markers have been linked to risk of prostate cancer. So today, we'd like to show you how we can estimate, assess the importance of the genetic marker in a cohort of about 3,000 men from a cohort established in France, the ProGen study. And for the purpose of the presentation today, we look for history, familial history of patients for prostate cancer, breast cancer, or other cancer. These are clinical data. Also obesity, because obesity has been related to risk of prostate cancer, and obviously genetic marker, single nucleotide polymorphism. We use for this presentation about 54 polymorphism, genetic variant. Each variant by individual define a genotype, and usually we have two allele. You are homozygote for allele or heterozygote. The problem is that the fraction of genetic risk based on this marker is dependent, in fact, of the population, because the frequency of the allele at risk in the population could be different between Asian, African, or European ancestry in the population. And we want to look specifically as a target node, target the question, the prediction of early onset prostate cancer before diagnosis before 65 years old, obviously adjusted the aggressiveness of the disease, because the majority of deaths due to prostate cancer are related to patients with early onset of this disease. So we have established this kind of data, your target node, the fact to have a prostate cancer before 56 years old, clinical factor, history of breast cancer, history of prostate cancer in the family, or to have other cancer different from a breast or a prostate cancer, but also body mass index. And you can see you have your genetic marker, which are referenced with this number. So it is a little bit confusing when you see this marker. The first step have been to define manually the definition of the node, and you can see a familiar history of prostate cancer. We have defined in the commentary of the node this, but for genetic marker, it is a little bit confusing, and we use generation of the definition with Elixir. We introduce a general context, prostate cancer and genetics. We use here the generative AI Claude for Sonnet, and we use for the definition consequence and cause, because obviously genetic marker is a cause of prostate cancer. It is also possible, as you can see, to use a file from a publication in order to precise the context of the study, in order to generate the definition. I use this publication based on the genetic marker and specific cancer deaths from mortality from prostate cancer. You can validate, obviously, all of the definition provided. And now we have generated for each genetic marker understandable and definition, which can be clearly read, and you can see that it is possible to have automatically the definition, a commentary about each marker. You can also publish a report about all the definition using Elixir tool. So the first step will be to use a supervised algorithm. We use naive Bayesian algorithm in order to connect all the nodes, and after we use also a Markov model, and you can see that it is very comfortable to can see for each node a clear definition from the previous commentary generated by Elixir. So the results have been presented here. You can see that familial prostate cancer is an important factor, independently of marker. Each marker have a weight, in fact, which could be different. Some have importance over others. It is interesting to see that with augmented naive Bayesian algorithm, you can see all the nodes and estimate the importance of each. In fact, you have a two factor, elasticity and contribution. Elasticity reflects, in fact, the direct effect. It is interesting because if you see the previous algorithm or graph, you can see that the Markov-Blanquet algorithm did not identify this strong marker, because this marker have a small frequency in the population. So you can see that this marker have a small importance, in fact, because it is relatively rare in the population, but at individual level, you have important risk increase. And in fact, direct effect, if you compare to epidemiological data, is relatively close to relative risk. And the contribution depend, in fact, of the prevalence of the marker in the population and give you the attributable fraction of this marker or factor in the population. Now we want to see if using generative artificial intelligence, we can have a network which could be close to your analysis on the real data. So we launch with Elixia two research models with ChatGPT and Claude Sonnet. We remove the target node because obviously it's too easy. All of these markers have been chosen to be linked to prostate cancer risk. So when we generate this network, interestingly, we can find that some of genetic variants are linked, in fact, to a body mass index, and other genetic variants are linked also not only to prostate cancer familial history, but also familial breast cancer or other cancer. When you launch this type of analysis, you have a clear commentary about the relationship between your marker, your genetic marker, and in fact, the veracity from the comment for the relationship between the two nodes, ici prostate cancer familial history and this marker. Obviously, we identify relationship not directly to prostate cancer history, but from BAMI with these two marker, genetic marker, and the relationship between the body mass index, risk of increased body mass index, risk of obesity, and obviously, consequently, risk of prostate cancer. In the same way, we can identify with that can related some markers which are involved in prostate cancer risk with breast cancer familial history. Usually this mutation such as BRC2, BRC1 are very well identified in prostate cancer, but it's more difficult to relate it for single polymorphism if this marker are only linked to a type of cancer or to another type. You can see that also the veracity here from the two analyses with the Claude Sonnet and the ChatGPT. So finally, we display this network, and we use ElixMap. ElixMap is a new tool associated to BayesiaLab, and you can see when you display your network in ElixMap, it is very interesting because we have a clear view of the relationship between the node. I put prostate cancer familial history in the center, but you have ici body mass index, also history of cancer and breast cancer familial history. And you see when you point out on each arc or node, you have a commentary, and it is comment provided by your motor. here is ChatGPT for this arc, but you have the same if you point or click on a node. So in conclusion, we can see that with this new tool associated to BayesiaLab, Elixia and ElixMap, it is relatively easy to define this type of marker. It could be also used for other genetic marker, because when you want to look for each marker in the literature, the function or the clear definition, you have a number time. You save time with this type of tool. We found that it is interesting to compare also elasticity and the contribution, because it is clear that they give a difference in terms of risk at the population level or individual level. And using this tool, we have also able to generate new relationship with ChatGPT or Claude Sonnet between some genetic marker and clinical factors such as history of cancer or body mass index, and which explain cross-link between this cancer in family, or relationship between obesity, risk of obesity, and risk of prostate cancer. So thank you very much for your attention. I hope I can show you that this new tool could be helpful in a different way, and particularly when we have numerous marker or information in your node in order to generate automatic definition.